In order to ensure that immune reactions are directed towards appropriate target antigens and not directed towards self-tissues, a number of mechanisms of immunological tolerance are employed. So in this mini lecture, I'm going to focus on what some of those mechanisms are and how those mechanisms break down to lead to the development of hypersensitivity reactions and autoimmune diseases. There are two kinds of tolerance that are referred to with regards to the adaptive immune system, and that is central and peripheral tolerance. Central tolerance occurs in the thymus. And as you'll recall from a previous lecture, we know that negative selection of self-reactive T cells occurs in the thymus, or in other words, T cells that might respond to tissue-specific antigens are eliminated. And those T cells generally undergo apoptosis during development. However, there's another phenomenon that occurs in the thymus in which some of these self-reactive T cells actually undergo differentiation into regulatory T cells, or Tregs. Now, in the periphery, circulating T cells may see activated dendritic cells presenting antigens. In this example, this is an activated dendritic cell also presenting co-stimulatory signals. And so signal 1 plus signal 2 leads to full T cell activation. But in settings of non-inflammation in which you're not infected, but you're being presented self-antigens, T cells receive signal 1 often in the absence of signal 2, and these antigens are presented to these T cells by immature dendritic cells, and this will lead to one of two outcomes typically. The first is this T cell may die, but the second is that this T cell may acquire a functional state called anergy, in which it has lost the ability to respond to T cell receptor signals. Another way in which T cells are tolerized in the periphery is through the action of regulatory T cells. So regulatory T cells specific for self-antigens may interact with immature dendritic cells, or they may even convert activated dendritic cells into immature dendritic cells. In turn, regulatory T cells can produce TGF-beta, transforming growth factor beta, as well as interleukin-10, and these cytokines have an immunosuppressive effect that suppresses T cell activation. Now, Tregs develop in the thymus, as I just mentioned, and so you have a constantly circulating population of Tregs at all times in your body, and it's thought that these Tregs are really important for suppressing autoimmune responses that might normally occur. Tregs are also induced during most immune responses, and these Tregs are likely to be specific for foreign antigens, but may play an important role in suppressing immunopathology that may result from out-of-control immune responses. Tregs are regulated by a transcription factor called FOXP3, and the reason I bring that up is that mutations in FOXP3 can lead to a disease syndrome called IPEX, or immunodysregulation polyendocrinopathy enteropathy X-linked syndrome. Yes, that is quite a mouthful. This is an autoimmune disease in multiple endocrine organs. It should be noted, however, that there's really no observation of a complete loss of FOXP3 function in humans. It's likely because, at least in experimental animals, a loss of FOXP3 entirely leads to overwhelming autoimmunity and death. So what happens when tolerance breaks down? And essentially, we've categorized immune reactions in which tolerance mechanisms don't function well as hypersensitivity reactions. And broadly, hypersensitivity reactions are when immune responses damage human tissues. And this can be a response to a foreign antigen. For example, here I've shown a red blood cell, and there's a well-known phenomenon in which penicillin can covalently attach to the surface of red blood cells. If antibodies develop to penicillin and bind to that penicillin, they may in turn induce the destruction of these red blood cells. And so even though the target of the antibody is not a self-antigen, it does lead to destruction of self-tissues. Alternatively, antibodies may be specific for a red blood cell specific antigen, a self-antigen, and this is a phenomenon that we term autoimmunity. So there are 4 types of hypersensitivity reactions that we will be discussing over the next little while in this course. The first are type 1 hypersensitivity reactions, and these are reactions mediated by IgE, and IgE is the principal driver of allergic responses. And so here I've shown a picture of a mast cell coated with IgE molecules waiting to find an allergen and activate that mast cell and induce an allergic response. Type 2 hypersensitivity reactions are mediated by IgG molecules, and this is when IgG binds to cell-associated antigens. And so as an example from the last slide, here's an antibody binding to a red blood cell and inducing destruction of that red blood cell. Type 3 hypersensitivity reactions are also mediated by IgG, but only when IgG is complexed with antigen, a structure termed an immune complex, which I've showed here. And these immune complexes can cause very specific types of damage. And again, these IgG molecules may be specific for a self-antigen, but often are specific for foreign antigens. But it is the presence of these immune complexes that can cause disease. And lastly are type 4 hypersensitivity reactions, and these are reactions mediated by T cells, either by T helper cells that produce cytokines such as interferon gamma and tumor necrosis factor, or by cytotoxic T lymphocytes. It's important to note that these are reactions, and for some immunopathologic diseases, multiple types of hypersensitivity reactions may play a role. So why do tolerance mechanisms break down? Why do we get hypersensitivity reactions? Well, first, genetics plays a really important role, and this can include HLA, sex, as well as other genes associated with regulation of the immune response. And most autoimmune and immune-mediated diseases are driven by complex inheritance. There's multiple loci involved and variable penetrance. There are some autoimmune diseases that are driven by a single gene mutation, and those are some of the genes that we've discussed in the past that you might consider to be involved very centrally in the development of immune tolerance. And this is things like CTLA4 and PD1 and FAS, FOXP3 and AIR, to name a few. Now, there are environmental factors that can lead to breakdown of tolerance, such as smoking or weight or age or diet. One of those key environmental factors is infectious history. And sometimes you can find a similarity between a foreign and a self-antigen, and this is called molecular mimicry. And it's thought that responses to these foreign antigens can then in turn lead to destruction of self-tissues. Okay. So as I mentioned, most inheritance for immune-mediated diseases is complex. And that's indicated in this figure, in which we see a number of genetic factors, multiple gene loci that contribute to disease, as well as infectious history, as well as environmental factors such as pregnancy or smoking or diet. And all of these things can combine together to induce an onset of an autoimmune disease. For complex inheritance, the locus that is most strongly linked to autoimmune disease is the HLA locus. And there's a number of diseases where HLA has been found to be linked to susceptibility to disease. Just a few of them are listed here. And I should note that for the vast majority of these diseases, class 2, MHC class 2 alleles are the ones that are most tightly linked to the onset of autoimmune disease, really highlighting the central importance of the CD4 T cell response in driving autoimmune diseases. But there are a few diseases that are linked to MHC class 1, and particularly this allele of MHC class 1, HLA B27. It is linked to ankylosing spondylitis, psoriasis, inflammatory bowel disease, and reactive arthritis. And so this is, whereas I won't hold you responsible for all of these other MHC alleles, you should remember HLA-B27 because it is used diagnostically for these diseases. And as shown here, is a really strong contributor to relative risk. There are some infections that trigger autoimmune responses and hypersensitivity reactions, and so I've just shown a list of infections and the types of autoimmune diseases that they are associated with. Again, don't go memorizing this list and the specific associations. I just wanted to make the point that infectious history is important, and infectious history on certain HLA backgrounds could lead to molecular mimicry and the induction of autoreactive T cell responses. And I also lastly wanted to mention that sex is a key variable here. And in fact, many autoimmune diseases, females are more susceptible. And this onset of increased susceptibility often begins in the third or fourth or fifth decade of life. The reasons for this are really poorly understood. And there's a few exceptions to that rule. For example, ankylosing spondylitis is more prevalent in males than females. But as you can see, most of these other diseases, including rheumatoid arthritis and lupus and Sjogren's syndrome, these are all more prevalent in females than in males. So here's a summary of the key points. Take a moment to go over these key points as you review the material from this mini lecture.